Thank you for coming in on such a gorgeous day to hear about my work around hematopoietic stem cells. So I'm an expert in manipulating hematopoietic stem cells and leukemia stem cells in a xenograft model. Because we can't really do experiments on you and I in the rigor that many of us would like to do. And I really care about what distinguishes them across our lifetime, and whether that heterogeneity that's inherent in our hematopoietic stem cell pool could underlie many of the age-related disorders that we are affected by. And so, just to get us all on the same page, I know a lot of you come from a blood field, right? A hematopoietic stem cell resides in the bone marrow. That pool has two remarkable abilities. It can self-renewal. It can also differentiate into all of the blood cells. But they are in polar opposites in terms of quantity, and those hematopoietic stem cells are quite rare. And in the context of the human system, from seminal work from Peter Campbell, Elisa Laurenti, and now others through normal hematopoiesis in various diseases, we know that genetically we only have about 50 to 200,000 of these clones in any given individual. But they have the ability to really proliferate so that there is such rampant daily turnover for immune cells, about 10 billion daily. In the context of the red blood cells and platelets that really prevent us from dying every time we bump into something, I mean, it's in the billions, right? Hundreds of billions. So with that enormous proliferative potential, they also go everywhere in our body. And this is, I think, when we're in the thick of things and we're really asking what makes a stem cell and how they go bad, we start forgetting that, outside of the bone marrow, they could be doing a lot of good or bad things outside of what, are the daily aspects of clotting, carrying oxygen, and immunity. Right? And so why does this matter? Well, in 2020, when the bulk of the story that you're going to hear about, it was a pivoting point for all of us. Of course, the COVID pandemic, but the one that really struck me was that that year, the World Health Organization reported that there were more people over the age of 60 than children younger than 5 years of age. We are living in a aging epidemic. And we also know from work in the last couple of years that HSC pool that is polyclonal, tens and hundreds of thousands of clones, by the time we get to age 70, there's 100 or less. There was this beautiful paper, a case study published outside from Spain, where the oldest living individual had 3 HSC clones. And you know what? All of them carried mutations, but she was healthy. So what is going on in our HSC pool that leads to a situation where we're contracting that pool, but some people have amazing health span, others do not? How can we parse that out? If we can do that, can we then figure out across our lifetime, where in this case, I'm showing you some data where Raffaella D'Amico, Serena Scala, and I did an aging compendium where this is bone marrow and just immunophenotypic analysis across lifetime. You see all these outliers, right? There are people that across this aging spectrum where, yes, there is a decrease of stem progenitor cells as we age. There is a bit of cytopenia, especially a decrease in B cells and anemia. Monocytes stay the same. In others, there is really high counts. But on the surface, they look healthy. Does that impact the blood production and risk for disease? And do we need to start thinking about both quality and quantity, and how do we measure that in our stem cell pool? And something that I've been really thinking about, we see these wheels a lot, right, is that within the hallmarks of aging and the hallmarks of cancer, There's one that has stood out to me and a lot of us. We hear about it a lot, inflammation. But that association of inflammation with aging and cancer is correlative to most extents and purposes, at least in us. In mouse models, it's beautiful work from many different groups. If over the last 10 years or so says the HSC pool, fitness goes down with age, it goes down with inflammation. But is this actually happening in us? How does this happen? And is the erosion of stem cell fitness perhaps in response to inflammation at the root of heterogeneous health outcomes when we look in the general population? With that introduction, I'm going to just get into a little bit of this concept of HSC heterogeneity. Looking at clonal hematopoiesis and how the response to something called inflammatory memory may really matter about how we rethink about what CH is, and to really go using the Xenograft model to ask about clonal expansion and the relationship with aging and inflammation, and then really asking about the translational implications of this story that I'm going to tell you about and the next steps. So this is one of the most pivotal experiments, functional experiments, I think, in the HSC field from the late Connie Eves. Her group just took single stem cells sorted from mice and then transplanted and looked across, 24 weeks. And the remarkable thing is, at any given stem cell will give a different pattern of engraftment. There is so much functional heterogeneity. Now, there are specific patterns. She brought it down to 4. In the context of the hematopoietic blood system, seminal work from Luca Biasco, we've done some. We know that in general in the human system, we have a population of cells that will give early engraftment and peter out. And there are others that are slow but persist. And we have some idea of what's going on there, but we have to reconcile this functional heterogeneity with the, up until recent, we still use to really that spiral stick model of hierarchical organization of hematopoiesis. And on top of that, in the last 5-10 years, and more now, right, every single paper has some single cell compendium. The single cell lens suggests, how do we reconcile this with this functional heterogeneity, where there is a continuum, whether we look at chromatin accessibility or the transcriptome? So measuring and interrogating that, but in a functional manner, is really what I've been going after for the last 15 years in the context of human hematopoiesis. And what we have found is, what distinguishes a stem cell is its ability to stay quiescent and maintain its self-renewal, but upon certain stimuli, it will activate, and then it has to make that decision. Does it stay a stem cell, or does it differentiate? And across sort of this just basic question of what regulates stem cell function, what makes a stem cell, I've been privileged to be part of many stories that says, it's a lot of things that integrate, and everything from cell cycle regulation, the epigenome is absolutely pivotal. But then other things that, when I set out to do some of this, Rob was on the same page with me, but we've turned, I think, the field, and others have joined us, that proteostasis, organelle biology, metabolic stress, and inflammation could all be tied together to help a stem cell decide if it's a stem cell or it's not. And in doing these projects, I kept coming back to why is there an underlying inflammatory program in normal cord blood stem cells? In fact, where we see, if we take these stem cells from umbilical cord blood, and we also know this is occurring in adult stem cells, they look, by all intents and purposes, quiescent. You put them in culture, within 6 hours, there's dramatic transcriptional change. And the 3 gene sets that come up are my most favorite lipids in the world, these sphingolipids, a reactome around TNF signaling, and essentially a fate decision around life or death. What is going on? Why is there inflammatory signaling here? And it took a while to decide to really go after it. I had to come up with a way of doing so. But when with that in mind, with all the work that was coming up around this phenomenon called clonal hematopoiesis, where somatic mutations of what were previously thought before seminal work from Sid Jaswell, Ben Ebert, and others have really said that these are found in high prevalence, as high as 1 in 5 over the age of 60 have mutations primarily in DNMT3A and TET2, but many others. These mutations prior to this were thought to be drivers within myeloid neoplasms, especially AML. So how can this be occurring? On top of that, it was shown that if you carry a mutation, even though you don't have cancer at the time, that it really increases your risk for mortality, increases your risk for hematological cancer over tenfold more. But you can see clonal hematopoiesis in the general population, in fact, in superagers, right? So, and the connection here is perhaps systemic. There is something around inflammation. And what has really become even more interesting, and there are very few hematopoiesis talks at AACR, but they have an entire session on clonal hematopoiesis. Is in recent years, the cancer community has found that not only does it raise risks for all these age-associated disease, if you are, in this case, a lung cancer patient and you carry a mutation in one of these clonal hematopoiesis genes, your risk of death is much higher than those that do not carry this. And more interestingly, they find that myeloid cells with a higher prevalence of the CH mutation, so the variant allele frequency that's in the blood, is found to be higher within the tumor. And it's the myeloid cells that are infiltrating. But we know myeloid cells are among the most short-lived of hematopoietic lineages. So how can this be? I have been quite struck by work from the incomparable Elaine Fuchs. Now 10 years ago, she just had this beautiful paper again in Science, showing that this phenomena upon wounding in mouse epithelial stem cells actually changes the stem cells, and they acquire something called inflammatory memory. And it allows them to respond better the second time. In the context of the skin, that means better repair, faster repair. In the context of the immune system, they call this trained immunity. And a lot of the work underlying childhood vaccination with BCG. And Maz Devarani, Mihail Natea, others have really shown that BCG, as well as essentially any inflammatory stimulus, can educate and cause a faster response from the myeloid cells through education of hematopoietic stem cells. But this is all in the mouse setting. So inspired by this, I set out to ask, is this a phenomena that occurs in the human system? And how does this integrate with the heterogeneity that we know is inherent in the hematopoietic stem cell pool? And by really modeling inflammatory response and recovery, can we ask if this is what distinguishes some individuals who are healthy from those that are not? And so we did this by doing these models, and it was a giant endeavor. 3 co-first authors, Andy Den, Murtaza Nagri, Asghar Jacobson, additional 3 second authors. There's so much computational analysis here that's going to blow your mind. I've tried to condense it. It will come out in a few weeks, but you're welcome to look through the 10 extended data figures and 5 supplemental notes, as well as everything else. But to boil it down, so Why did I do this? Well, for over a decade, we knew that there was heterogeneity. And when quite a few years now, we did this experiment where we just purified CD49F long-term HSCs from umbilical cord. And we asked, well, let's look at the single cell level if there is heterogeneity in those quiescence programs, inflammatory programs, and others that we've been, dancing around for a long time. And it turns out there are. And in fact, they're polar opposites from each other. Using machine learning here, we're just showing you that there are some that are highly quiescent and there are some that are highly inflammatory, especially around TNF signaling. And this is happening in neonatal cord. It is, of course, not immune naive, but it's the cleanest we can get in our system. So if we take that cord blood, enrich for their stem progenitor cells, and put them into the xenograft, and then ask, how do they respond to acute inflammatory stimulus? I had lots of work to draw from the mouse system to decide, but what I did decide to do was to ask not just about recovery from bacterial infection, but human TNF, because it kept coming up again and again, and there weren't comparable experiments for human TNF other than Manuel Pasagay and Masa Yamashita's data, and it was very compelling to me. I wanted to ask, is this the same in the xenograft? Can we model it? And to show you, we did this at an early point in transplantation, because everything is undergoing xenograft and transplantation, so that itself is stressful. So we did it at a time point where we know the stem cells are cycling, from a paper that Lisa, Catherine, and I did in 2015. Also at 20 weeks, when we know most of the stem cells have gone back to dormancy. In both cases, blue is TNF, red is LPS. You can see that engraftment marked by CD45 as well as CD34 to mark the stem progenitor pool, there is a decrease. Okay, that's great. They respond to acute inflammation, things go down. This is what happens in the mouse. Now, what's happening to the cytokine environment? Can we recapitulate the cytokine storm that we see in us? So we did the plasma cytokine profiling with a company where we can look at one cytokine here, TNF, and we used LPS to show, yes, we can see it go up. We looked, in fact, at 23 human cytokines, 18 mouse cytokines. In the xenograft setting, we are creating a cytokine storm situation. And that graph can generate human cytokines in abundance. Okay, now, this is the real experiment. This was actually the first experiment I did, which was to take that same xenograft model, but now we do one challenge, and then we take some of the animals through, we repeat the challenge, and then give them a chase period of 10 weeks. And with a single challenge, yay, they recover, because we don't die and the animals don't die either. Wonderful. Everything, at least at the level of graft, is the same, but lo and behold, with a second challenge, now it looks like, at least grossly, like acute inflammatory stimulus. And so there's durable change. And this has to come from the stem cells, because that's at 20 weeks. We've done lots of work. It's only a stem cell graft. So now was the big thing. It's just a lot of money and technical issues here, but we sorted 427 million cells from all of these animals, put them through, purified out those rare stem progenitor cells, the 10x single cell multiome, so that we can look both the chromatin accessibility and the transcriptome. Andy has this beautiful paper. He had to create a reference map so we can call all the populations out of this. And that set of cells are both stem and progenitor cells. For anyone doing xenograft analysis, marker fidelity is not as tight as out of primary sources, but we can use this reference map to call the HSCs and everything else, and to boil it down, this UMAP embedding of all three conditions, well, yes, there's a continuum here in blue of a stem cell population and everything else, but we got this side population, another HSC population marked in green, and some progeny. And by all intents and purposes of signatures that are out there, including ones we generated here at the transcriptome and chromatin accessibility level, these are stem cells. They're bona fide stem cells by these molecular signatures. But unlike out of neonatal cord, now out of the xenograft, this second HSC population are actually both inflammatory and they are quiescent. So what's going on? So to really ask, and this is just to show you that, yes, there are two HSC populations, but they differ once from the ones that recover from TNF or LPS. So we went after the gene regulatory networks using this machine learning tool called Scenic Plus, where we can really connect transcriptional changes and epigenetic changes together. And all this fancy stuff that I don't understand, but fortunately Andy's a whiz at. And what it boils down to is that one of this, the HSC1 population, when he looks at comparing PBS to in this case TNF, he also did it with LPS. You look in the paper and look at all the GRNs. There are essentially no significant changes. The changes are underlying HSC2, and it's NF-kappa B and AP1. Phos and Jun that has now come up in pretty much every case looking at inflammatory memory, from Elaine Fuchs and very recently from Jason Boehm-Rostrill's group, pancreatic cancer. Nicola Villay in 2019 also showed it as well. So there's something about this program, and it's durable. So there are two populations. This is just a plug for a paper that's going to come out. We spent 3 years. I'm not done yet, but so far we have not found any prospective isolation markers for HSC-IM to distinguish from HSC1. But one of the top candidates is this calcium exporter called ATP2B1. There is an antibody for it, and we spent a couple of years characterizing it, because it turns out that we can separate CD49F long-term HSCs and fractionate it into different populations. So CD9F positive cells can be positive for ATP2B1 or negative, and we did primary to secondary transplants. The double positive cells here are more functional, and in fact, even if you just take them and then put them in, we have not been able to, at least with this limited set of mice, all of them engrafted. This seems to be marking a superior new population of HSCs, which we hope will be a useful tool in various human disorders. So that's coming out in the proofs we looked at, so it should be out online very soon. So no markers. But what we have found is that there are actually two HSC subsets. When you put in an empirical model to really ask about stress recovery, both from inflammation and proliferative stress. And only one of them, this HSC2, which we named to HSC inflammatory memory, has durable transcriptional and epigenetic changes. So who cares? It was done in the xenograft, right? So what does this mean? So we started first at what was is the best known situation about memory, T-cell memory. And there's this beautiful real-world data set where volunteers took yellow fever vaccination. They also drank deuterated water, and the investigators were able to get both naive, effector, and long-term T cells from some of these volunteers 4 to 13 years out. And they did 10x motiome as well. So we didn't have platform challenges. And Andy generated, among the various ways he looked at it, was to create a long-term stem cell signature, memory or T-cell signature, and it shares many properties with our HSC-IM. He also generated a chromatin-based signature, as well as the top 200 genes. You'll hear me talk about this one a lot following, is what we call the HSC-IM signature. And he showed that both at the chromatin level and the transcriptional level, there are shared properties between our HSC-IM state and long-term T cell memory in people. So, okay, I care about aging. So we pulled together from 41 donors, including 6 that we did in-house, and this represented 23,000 transcriptional HSCs. And across these data sets, we asked, does HSC-IM enrich for age HSCs? And these lollipop plots looking at normalized enrichment score, I hope you can see how strong it is. Yes. Okay, accumulates with age. Then we went about it and looked in the scenario of, yes, aging. We created aging magnet signature. I will come back to one of them. It's called CD83. And that in itself might be a good resource for some people. It is enriched in HSC-IM out of the Xenograft. We use this beautiful paper from my colleague and now friend, Stephen Josephowitz. Through this, he shared this data before it was published, and we looked. And in the context of post-COVID recovery in individuals that were hospitalized for severe COVID infection also is likened to what we see out of the Xenograft. And what is most intriguing to us was in partnering with Parrish Vaez, where they have this data set that I'm going to talk about more, where we can look at stem cells that are mutated versus that are not from individuals with out of bone marrow. We could generate a CH-associated signature. It's enriched in HSC-IM. As part of the long process of getting this out to press to you guys, we were asked, well, all those scenarios are across aging, right? Is it specifically around inflammatory stress? So we went into sickle cell anemia, where it's associated with a premature aging inflammatory state. And there was this, fortunately, a paper that just came out recently where we can interrogate and ask. And we found that those pediatric patients that have sickle cell disease does in fact have an enrichment of HSC-IM versus HSC-1, but it's heterogeneous. So you can appreciate that. And if we have more data and we can go into each individual and ask, could this be a biomarker perhaps for things like gene therapy? We don't know, but certainly it's there, but it's heterogeneous. Again, this is all correlative, right? Are there really 2 HSC populations in us? So we turn back to this clonal hematopoiesis paper that we collaborated with Parrish on, where there was 13 donors, 14, so often I don't even remember. And we, Asgard and Sven, did target-seq, this protocol from Adam Mead, and they made it better. And they were able to look at the transcriptome, the immunophenotype, as well as do genotyping on this. And we did a bunch of work to say, hey, mutant stem cells out of people with CH actually have attenuated response to inflammation. But that data set also allowed us to do machine learning to ask within there, are there 2 HSC populations? I can hope you appreciate, yes, there are. And when we took our HSC-1 and HSC-IM programs from the Xenograft and projected onto this, that ROC score, they're essentially identical. So yes, this can happen in people. And now it gave us the possibility, knowing that HSC-IM, that state, is created, in fact, in response to recovery from acute inflammation, to ask, is this occurring in these individuals? And perhaps to ask about what comes first. Is it an inflammatory environment that's generating the state? And this is what we did here. And we can see that when we compare the stem cells with CH from not, that it is there. And so, in fact, whether you're CH mutant, CH wild type, when we compare it to the controls, you have, especially of DNMT3A, an equal enrichment, less so in the context of TAT2, and we can discuss why that might be the case. But this is suggesting that, hey, this state may be preceding any selective advantage of that CH mutation when it first forms. But that mutation could actually be changing how the stem cells respond to the inflammatory environment. And we have evidence of this because we can see that there is a downregulation of, among other things, TNF alpha V and NF kappa B signaling when we compare the mutant stem cells to the wild type. So if this is the case, then we would expect that there would be decrease in aging signatures. We do. We also see that there is a nice correlation of enrichment for that HSC-IM state relative to the clone size across this limited data set. So is this really occurring? Well, we can start asking about the outcome of HSC1 and HSC-IM in this cohort. So that's what we did. The first thing we had to do was to take that set of samples and classify what proportion of their stem cell pool is HSC1 and HSC-IM. This is what shown here. Some of them are HSC1 dominant, some of them are IM dominant, some of them are mixed, and it's not dependent on mutation status. So now we have this scenario to then do pseudotime analysis. And what Andy did was to just look at the wild type cells and across from stem cells down to progenitors, who has better output. And I hope you can appreciate the green cells, those HSC-IM, have decreased output in this analysis relative to the HSC1 enriched cells. More importantly, when we look in either HSC1 dominant or HSC-IM dominant, now we can ask, does the mutation do anything? So if there's a limited output, if you're coming from an HSC-IM state, you would imagine that now you should have increased output, right? If you carry a mutation. In fact, that's what we see here. So this suggests something around clonal advantage, that the mutation is taking advantage of the inflammatory aging state that is occurring, right? And it's not positive clonal expansion. In fact, it could be a negative selection of the wild type stem cells. So then we asked, is it heritable? And in that CH data set, we have we have cells down to few, but we have mature cells as well. And Andy generated a signature of 257 genes, if I can recall, and then asked, looking at HSC-IM versus HSC1 dominant cells, to taking that signature across the hierarchy, that there is enrichment of IM signature all the way down to the monocytes. There's also a retention of this quiescent state, at least to GMPs, and it's still there a little bit in the monocytes, and they're inflammatory. There seems to be some transmission of this state down to the progeny. But this is a limited data set, and every individual, we don't know the exosomic circumstances of their life. There could be lots of potential for this to occur. So we went back to the Xenograft model, where we sorted out the stem progenitor cells. We also sorted out, within that 427 million cells, the mature myeloid cells and the progenitor cells. And we used a trick called supercell that for the computational biologists, you're typically using this pipeline to call doublets. But it can see small nucleotide polymorphisms. And this set of experiments was across 3 different pools of cord blood with over 62 donors. So we're able to classify some genetic clades, clonal groups, and ask, again, in the same way we did with the individual bone marrow samples. Some of them are HSC1 dominant, some of them are HSC-IM dominant, some are in between, but this lets us essentially have a trick of genetic lineage tracing. And from this, we know that IM cells, stem cells, have enrichment, and they will pass down to monocytes. And if we then go through the entire hierarchy from stem progenitor cells through the precursor state, including B-like cells to monocytes to DCs, we can see that there is actually transmission all throughout the state if you're IM versus 1. So they're transmitting these programs. Again, this is great. It's in a state where everybody was in the mouse, right? So there's suggesting that there is something to heritability, that it's intrinsic. But does this have any application? So we partnered and really took advantage of a cohort epidemiological study in Canada called CAMPATH. And this is from one of the provinces, mine in Ontario, where Philip had, from individuals that had volunteered to enroll in this, they had a blood sample. And they used a mortality risk score based on just common blood parameters that's been out there for over 10 years that says that depending on how these parameters are, it's an Intermountain risk score. If you are greater than 5, your risk for 5-year mortality is much higher than if you're below 3. And they classify these 400 individuals across the lifespan from their 20s into their 80s, and they fractionated across, some that are young and low risk, or high risk and young, or some that are age over 65 that have a low risk and high risk. And we asked, does this in any way correlate in this Punnett square here? And what we found was in the young individuals, there was an enrichment of this HSC-IM program. This is no stem cells here, right? We're just asking, could this be a biomarker? And we are finding that there is this correlation with all-cause mortality, and we're seeing it most interestingly in dendritic cells, B cells, and NK cells, which, are becoming of great interest in the context of immune signaling, immunotherapy, the immune microenvironment. Could that stem cell signature be passing down to various immune cells that are infiltrating into tumors or in other diseases that can be leading to the heterogeneous outcomes? So with this, I would like to summarize. We found using just xenograft models that there are 2 HSC subsets. One of them retains molecular memory upon prior recovery from prior inflammatory stress. It's quiescent, has limited hematopoietic output, but with aging where somatic mutations build, there's now a selective pressure within HSC-IM for these CH mutations to relieve that differentiation decrease, to allow for increased differentiation. And this could be one reason why DNMT3A is so prominent, because if our stem cell pool is so quiescent, we're not making very much blood. So that could be the selective pressure. They transmit these HSC-IM, their pro-inflammatory properties to downstream progenitors, as well as it seems to be all immune effector cells. This has implications for inflammatory associated diseases across lifespan. So what's the implication here? One way to interpret this is actually those red dots, that CH variant allele frequency that we see increase, may actually be selective advantage. We're not seeing those clones that may have become very, very dormant. And that this is perhaps due to that inflammatory niche in each individual. If that's the case, then we need to think about targeting the inflammatory niche, and perhaps we can take lessons from obesity. Is there something that we can use to target inflammation and mitigate CH expansion? Well, everybody and their mother. I was reading an article, opinion piece in the New York Times right now. It appears that the bros on social media that are down to this case study of an individual had concussion are taking these, whether off-label or on-label, and some of the indications are outside of weight. It seems to be for their anti-inflammatory properties, and we know that obesity-induced inflammation exacerbates clonal hematopoiesis. So could we co-opt the properties of GLP-1 to mitigate inflammation? And luckily, Daniel Drucker, one of the prominent experts on this, is literally next door to us at Princess Margaret. And he had this beautiful paper from 2023 where he showed that is on target with GLP-1 receptor. You require it, but you can dampen TNF output with GLP-1 out of these syngeneic mice models. And recently, Stephen Chen and we helped them show that using another common diabetic drug, metformin, reduces competitive advantage, at least for the NMT3A AA2H. So we decided to think about this, and at the time we wrote the grant, this also came out. There were multiple epidemiological studies saying that in diabetic patients, that the decrease of there was a decrease of colorectal cancer, of liver cancer relative to insulin in these cases, as well as blood cancer. So we created this team initiative, and remarkably, CIHI took it for cancer prevention, and we've started doing pilot studies to really ask, can we alter clonal VAF size in this case in the mouse model, where we took TET2 mutant cells transplanted 1-to-1, and we've been giving them daily injections of semaglutide between 4 individuals. There's a lot of mouse handling, and we're seeing some efficacy at a relatively low dose. So that's heartening to us, and we're doing more pilots and really going full force now on this. And I'm a hematopoietic stem cell expert, right? I have this acute inflammatory model. I'm not going to look at that first. I want to see if there's a cytokine change. So we did this experiment. We piggybacked on my acute inflammation model, and we asked, can we see changes with pretreatment of GLP-1 followed by LPS stimulus relative to PBS control? And we used a marker that we have found coming out of the HSC-IM story. It is we found to be exquisite biomarker in long-term HSCs, highly, highly sensitive to TNF, as shown here. And so when we did this relative to animals that were transplanted and then just treated with vehicle or with LPS, either with another vehicle LPS sub-Q or GLP-1 pretreatment, we're seeing a decrease, not just in CD83 as acute inflammatory marker, we're seeing a decrease in TNF as well. So there could be something to this. So we've really gone full force now. We're going across the next 5 years to really ask mechanistically, can GLP-1 or some of the other agents really target CH, variant allele frequency in syngeneic models? If so, how? We're doing Xenograft models with CRISPR mutations to DNMT3 and TET2 in the human setting, and we've partnered with an endocrinologist around Toronto to do observational study from the endocrinology clinic to see who has CH, and then they're going to be funneled into our CHIP clinic, and to really ask, can we guard against clonal hematopoiesis with GLP-1 receptor agonists? Thank you so much for your time. And if you're almost there I am still looking for postdocs. Come and talk to me. Thank you.
Thanks, Stephanie. Really terrific talk. Really cool. Thinking about how our HSCs are changing. And so I've got a couple, I have lots of questions, but we'll start with one or two. With the HSC-IM, why, as we get older, aren't we all just filled predominantly with HSC-IM, right? We have lots of acute inflammatory stress over the course of our lives, and yet you show this like remarkable heterogeneity of that heterogeneity, right?
Yeah.
Where some people are
Yeah.
still dominant with
I'll go back
HSC-1s
to-
and some with
Yep.
these HSC-IMs. And
Yep.
any explanation or ideas?
No explanation. So this is where I'm continuing the partnership with Philip, and hopefully Cancer Research UK will give us a bunch of money to really, because it's hard. It's hard, right? This is the only study we have right now where we can really ask, and also that around mutations, we could potentially, with all the work that we've shown, mutations don't need to occur. I mean, KJ Patel, with his beautiful paper that just came out in Cell Stem Cell, says there are other ways to get clonal expansion that is not somatic mutation driven. So if we can use the IM signature or other ways to boil it down, use it as a biomarker, can we perhaps not do it on stem progenitor cells, but just out of our blood, given that it's transmitting, and classify people as one or the other, is I think something to be of use. But it doesn't answer your question. The question is, why is it there in the first place? Where I was very careful to say that neonatal cord is the most immune naive we have, but it's not. There is plenty of inflammation in utero. In fact, especially with all these cases of preeclampsia and others, could there be ready selection in the womb? I don't know, but I would love to find out. I've been trying to track down individuals, obstetricians who could give me some samples, or at least out there. But that's something that I really wonder about.
Yeah. So are we predestined to fall into one of these categories?
I wonder. But the flip side is that's why we're going after, we sold the GLP-1 receptor agonist story as cancer prevention, but it's really to see if there is a predestiny. Can we swap, right? Because there's a mixture. There's never not any one or the other. I mean, this individual had the low, like it was 98%, but we always see the other population in every scenario we look at. So is it just the time when we sample them? Oh, how dynamic it is. I think that is something we don't completely understand, but we have some evidence, at least from 5 of these individuals. I didn't show it because our reviewers asked. We transplanted, and it is transmissible.
And you sort of focus on the downside of these HSC-IM. Is there a good side to them? Like,
I sell it as such, but I think that it's actually pivotal. I think I think a lot of the anti-inflammation treatments, at least after specific cytokines, we don't have enough evidence to go after them, because a TNF signaling, at least on the human side, it's an underlying feature of what makes them stem cells. They have to respond quickly. I think, especially with the prevalence of DNMT3A, I think it is there is, nature is smart. Nature is doing this to allow for that lifelong blood production. And if we're always responding to every agent coming through like we are neonatal cord, will be gone. Our blood source will be gone, right? So I think it is the training at the level of hematopoietic stem cells is to go more dormant, but as we are aging, our population, expands. But maybe that's why we're seeing clonal hematopoiesis. That's the flip side of the evolutionary selection. Yeah.
Beautiful work. I'm really curious about the kind of relation between the cytokine signatures and cytokine storm and inflammatory IM HSC and cytokine storm in patients receiving CAR T therapy. Are there ways that these signatures could be leveraged or as biomarkers of predicting inflammatory reactions in patients in the clinic like that?
Mouse and human are interspersed here. There, at least with LPS, all goes down. We among the supplemental data, we actually show at 20 weeks we capture the bone marrow aspirates, and a handful of them are still up. even the PBS setting. So transplantation is a little bit stressful. So which of these are important? This is a xenograft model, right? We can only say that the grafts are making these and they are responding as they should. As you say, it's critical. Whenever I talk about this, or I see clinical colleagues are on trials, I'm like, do you have plasma bank? Can we stratify and start asking, with outcomes? We may or may not need mutation. I think we just need outcomes. And then can we go back to the patients and sample their blood to ask, how much of this is from their blood and not the tumor itself? Could one, I'm so HSC centric, right? Like we love the blood, but one possibility from this is, a certain amount of this is, and the immunotherapy revolution has said so much of cancer could be just the immune system not responding the way they should. Right? So the other part of the immune system is they shouldn't be acting up, and at times they should be flexible. I think our immune systems as we age become inflexible. They're not toggling back and forth as quickly as they should, and trying to figure out exactly which cell types, which of the signaling pathways. This could be which level that's at, I think is taking me down to speak to a lot of immunologists. Yeah.
I'm wondering, in the this sort of chronic expression of inflammatory genes, have you looked whether or not there's any negative regulator of expression of like AP1 mediated expression of these genes, which is lost in the IM cells?
I'm looking for a computational postdoc to really, really delve into. In our chromatin data, we used it, but to be perfectly frank, we didn't go into it as deeply as we could have. Part of it's hard, and we're lacking a critical component, methylation, right? Like I've been thinking for years that this was required, but we needed to get this across the line. But also, in the context of these rare stem cells, like it requires some potential single cell methylation, or at least key marks. Now, from Elaine Fuchs's recent paper, it's narrowed down to a handful that is, I think, doable to at least cherry pick and do in the human setting. So I'm working with some colleagues on that. But yes, I think that's the wave. And if I may add, I think part of it is actually the binding partners. In all cases, AP1 is there, but at least in pancreatic cancer versus the beautiful study from Rostrell's group and this latest one from Elaine Fuchs, one's mouse epithelial, normal epithelial stem cells. The other is a KRAS, colorectal cancer model. Oh, sorry, is it APC-Min? I now forget what the driver is. But it's the transcriptional modifiers, the transcription factors that actually modifies and goes and says which of the open areas are transcribed further down and respond quickly. And in that case, I think it's going to be tissue specific.
Hi, thank you for your beautiful talk. I have a very basic question about the origin of the IM2 type, like of hematopoietic stem cells, because they have like the similar functions with like the type 1 and regulatory programs that are like equal. Have you done some like transcriptional trajectories, like analysis to see where these cells are coming from and the origin of the cells?
If I understand the question, we have. I mean, that's what all this pseudotime analysis is, and that's just a couple of examples. We've also done it across everything from Vijay Sankarant's his mito tracing paper, where we also have done this sort of thing, and at least with the data sets that are available out there, we've done it. Have we done actual lineage tracing? We tried. Yeah, marking with something like Larry, and then being able to transplant and pull back clones is very, very inefficient in the human setting. That doesn't mean that I'm giving up. It's gotten better. I keep in close contact with some of the best out there. Alejo Rodriguez Fraticelli is great at this. I keep and you know but there we might need other tricks to do it. The other issue at play is all of the lineage tracing right now, at least in the hematopoietic system. Primary requires lentiviral barcoding. That's very stressful. That with my CD83 marker, anytime you put any stem cell into culture, starts lighting, and you're selecting. Yeah.
Great talk. Quick question. So HSC-IMs, like, obviously nicely showed many inflammatory signatures obviously are associated in them. I was curious to try and sort of bring it back to some of your earlier work and sort of how you were mentioning you and Rob were like the forebearers of sort of looking at metabolism and HSC, et cetera. Yeah. So I'm wondering what other stuff was associated with HSC-IM, like any sphingolipids or proteostasis, or I'd also be curious about ribosomal biogenesis factors.
Yep. So that ATP2B1, that new marker, it's a calcium exporter. We trace the mechanism for its increased functionality to endolysosomal functions. They're marking the cells that have the most nuclear TFEB. Yep. So at least there, that's a big part of it. That dormancy state is in fact probably upregulation of a lot of the programs we know that retain sort of that inherent self-renewal. Now, your main question around, my beloved sphingolipids, I put them aside for now almost 6 years. That wasn't the intention. I thought I would just have a signature and I would use this set of xenografts, and those are ongoing. I'm going back to. I didn't talk about it because it's published work, but especially this blood cancer discovery paper. I did all this. I gave almost 9 years of my life trying to disentangle sphingolipid metabolism and AML, and it was all around inflammation. So that's one of the reasons why I need a postdoc. We have a system, we have a phenotype, and now we need someone to really do it. Thank you.
Thank you so much for your great talk. Your comment briefly about sickle cell disease kind of brought up some questions for me clinically. I was wondering, within this HSC-IM, if it's something that could be used to stratify the cells that are edited for gene therapy, or stratify cells that are transplanted in stem cell transplant.
So we have an ongoing collaboration with Giuliana Ferrari, not in sickle cell, but in beta thalassemia in the context of gene therapy editing and outcome. Some of her patients responded to the editing successfully, and others did not. And we can use the HSC-IM and other genes encased in that to sort of, we're trying to disentangle it. And some of it is programs we're not, there's an ASH abstract out there. So HSC-IM is not one single state even there. We can see that a subset of cells are reliant on more interferon, and another subset is reliant on a TGF beta program, and what that may mean, we don't know. But across various diseases, across aging, we can further classify some individuals that have a stronger IMA versus IMB. That one I don't know how long it'll take to disentangle. But thank you.
Thanks, Stephanie, again for a terrific talk.